All right, so we're going to finish this up here real quick. So independent assortment, the idea that the genes will separate, or the chromosomes will separate without worrying about what chromosomes they're getting sorted out with, okay, that idea, and the process called crossing over that happened during meiosis results in that genetic diversity, okay, and then you get the combination of the different gametes that produces even more genetic diversity. So independent assortment is the allele pairs separate independently of each other during meiosis, okay? And then crossing over is the exchange of chromosome segments between homologous chromosomes. Now this is the part that gets a little bit weird. It happens during prophase 1. Okay, so when the homologous chromosomes are starting to condense, they'll actually kind of start pairing up, and that's when they trade off genetic information. So it results in new combination of genes, and it results in each of the gametes being produced by that one individual being completely different from the other gametes. All right, so let's look at crossing over. So two homologous chromosomes will pair up with each other during prophase 1 in meiosis. At this point, they will actually, well, let me get my laser pointer going in here. Okay, so at this point, the chromatids are actually really close to each other, and so the segments will actually cross. Okay, they kind of get a little tangled up with each other. Now, at this point, you will actually have, they get a little bit confused about which section comes from which chromatid. So they will switch information. Okay, so you get a segment that breaks off and reattaches to the other homologous chromosome, which then gets its own new piece of DNA. Okay, so this process again is really strange, and what you'll note is that you have two sister chromatids on each chromosome, but they don't always have to both go through crossing over. Sometimes it can be just one of the chromatids on the chromosome will undergo crossing over with another chromatid on the other homologous chromosome. Okay, so basically this chromatid on this left side of the purple one doesn't have to undergo crossing over with the green one on the right. Okay, it's just one of the chromatids from each chromosome will. Okay, and that means that this chromatid on the on the right of the purple one here that has the green segment on it is now genetically unique from the one on the left, the purple one on the left. Okay, so in that way you end up with gametes that are going to be different, genetically unique. So chromosomes contain a lot of genes, so we're also going to talk about something called gene linkage real quick. It's a relatively easy concept, kind of logical concept. So the farther apart two genes are located on a chromosome, the more likely they are to be separated by crossing over. Though genes that are located really close together on a chromosome tend to be inherited together. So that is known as gene linkage. It's a breakable link because it is possible that they will be separated, and you do see that sometimes, okay? But this is kind of like, if you think about Irish people, you usually think of pale skin, usually blue eyes, or green eyes, and red hair. Okay, so those traits, especially the red hair and the pale skin and freckles and all of that, those traits tend to be on the same chromosome and really close together. Okay, so they are considered linked. You'll see them show up in that combination quite often. And then if you see, an Irish person that has, say, blonde hair, okay, there was a separation that occurred there, and that was kind of a little weird, right? You'll also see this with a lot of people who have darker hair. Oftentimes they will end up with brown eyes. It's not an always kind of thing, but oftentimes you will see that. If you think about what Germans look like, generally it's very fair hair, like blonde hair and blue eyes. Okay? And not just because of the whole Hitler Aryan race thing. It's actually kind of what was I mean, there was a reason why he said that was the perfect race. It's because that's what a lot of Germans actually had naturally. So genetic linkage actually allows the distance between two genes to be calculated and mapped. So this is how they come up with kind of the locus of the different genes. They figure out the relationship to other genes on the same chromosome through this genetic linkage. So this is kind of what it looks like. Basically, you can say that A and B are linked because they are incredibly close together. So they are generally going to be inherited together. Okay, so every once in a while you might see a separation between the two, but it's going to have to be a very precise separation because they are so close. Okay, so it's going to be very, very rare. And because it's that rare, you can actually tell that they are incredibly close together. Okay, so A is not linked to C, however. They are on completely opposite ends of the chromosome, and it's actually very likely that they will be separated during crossing over. Okay, and same goes with A and D, and then also with B and C and B and D. Okay, but yet again, you've got C and D are right next to each other, so generally you would consider them linked. Okay, because most likely they are going to be inherited together. Okay, all right, so that is it for this chapter.